Hello friends, welcome back to my channel. In this video, we'll be talking about the synthesis of creatine. Okay, so the synthesis of creatine is an inter-organ process. It is an inter-organ process, meaning that one of the reaction required for the creatine synthesis it happens to be in the kidney, and the other enzyme or the next reactions that is catalyzed during the synthesis of creatine happens to be in the liver. Okay, so in the kidney and the liver the creatine is synthesized. Okay, and the enzyme synthesizing the creatine are AGAT, which is arginine, glycine, amidino transferase enzyme, which is mostly expressed in kidney, pancreas. Okay, and the next enzyme is GMAT, which is guanidinoacetate methyl transferase, which is expressed in the liver. Okay, so the process of creatine synthesis is an inter-organ process. Okay, and there are two enzymes or two steps in which the creatine is synthesized, and there is requirement of different amino acid like arginine, glycine, and also methionine that is required for the synthesis of creatine. Okay, so the sequence of reaction, okay, or the enzymatic reaction, I'll be discussing it now. Okay, so as I told you, the first reaction, okay, during the creatine synthesis happens in the kidney. Okay, and the enzyme catalyzing the reaction, the first step in the creatine synthesis is AGAT, which is arginine glycine amidino transferase. Okay, so what happens is there is a transfer of amidino group from arginine to glycine, which result in the formation of guanidinoacetate. Okay, formation of guanidinoacetate and ornithine. And this reaction exclusively happens to be in kidney. Okay, the next step in creatine synthesis is now in the liver, where this guanidinoacetate is methylated by the enzyme guanidinoacetate methyl transferase enzyme, guanidinoacetate methyl transferase enzyme. Okay, and this guanidinoacetate is now converted into creatine. Okay, so and in this reaction, this enzyme require the methyl donor. Okay, and that methyl donor is S-adenosyl methionine or SAM. Okay, so in this reaction, in the second reaction, after the transfer of the methyl group to guanidinoacetate, this S-adenosyl methionine is converted into S-adenosyl homocysteine. Okay, so in this way the creatine it is synthesized with it is an inter-organ process, it requires the activity of two enzymes, which is AGAT and GAMT, guanidino acetate methyl transferase enzyme. Okay, so this creatine, okay, the creatine that is formed during the creatine, after the synthesis of this creatine, okay, the 95% of the creatine that is synthesized is transported to the muscle, and the rest 5% to heart, brain, okay, right? And this creatine, it is stored in the form of creatine phosphate. This creatine phosphate, it is a high energy compound that is exclusively used during anaerobic respiration. That is anaerobic respiration. Okay. So this creatine phosphate, okay, the creatine phosphate, which is mainly stored in the muscle, okay, which is mainly stored in the muscles, contribute as a phosphate donor, okay, during anaerobic respiration. Okay, anaerobic respiration means when the rate of consumption of ATP exceed the rate of production. Okay, so the creatine phosphate, since it is a high energy compound, it donates its phosphate group to ADP, okay, that is converted into ATP, which is utilized by the muscle for the contraction purpose. Okay, thank you.